When you have completed the staining of your bean beetle ovarioles, you'll want to transfer them from the staining dish into a microfuge tube containing PBST. You'll want to use the blue tips, which have a larger bore, and the P1000 to suck up your ovarioles and then place them into the microfuge tube. After you have placed them into the microfuge tube, you'll want to place the tube under the microscope and check to make sure that your sample is in the bottom of the tube. After you have confirmed that there is a sample in the bottom of your microfuge tube, incubate the sample for 10 minutes in the PBST. Following the incubation, you will remove the PBST, leaving the sample behind in the test tube. During the last step, when you're removing the last little bit of PBST, be very careful that you do not accidentally suck up the sample into your tip. You can actually check the tip to make sure that there are no ovarioles in the tip. Once you've removed all of the fluid, check to make sure your sample is still in the bottom of the microfuge tube. Once you have confirmed that your sample is still in the bottom of the tube after you have removed the PBST wash, you will then carefully remove 100 microliters of floor mount. Floor mount is viscous and you will want to do this very slowly, avoiding bubbles. Take the 100 microliters out using the blue pipette tip with the larger bore, the P1000 pipette. Add your 100 microliters to your sample in the microfuge tube. Again, confirm that your sample is still in the bottom of your tube. Now you are ready to make your slide. You will again use the larger bored blue pipette tip with the P1000. Carefully remove the sample in the floor mount and place it onto a slide. If you do get bubbles on your slide, you can just gently suck up the bubbles into your pipette tip and remove them. You want to avoid bubbles. You don't want them in your final slide prep. Once you have placed your sample on the slide, place a cover slip along one edge and gently lower the cover slip onto the sample without getting any bubbles. Then place the slide under the microscope and check one last time to make sure that you have a sample on the slide. You have now completed the staining procedure.